So good evening to all the participants in India and good morning to our speaker. So indeed, it's my great pleasure to welcome you all for this IISc TIFR webinar series. So today is day 2. Yesterday you heard Professor Phil Kukura, a wonderful lectures on single molecule detection and emission properties. So this webinar we started sometime in the month of November 2019. That's where we were in the middle of the pandemic. And we increasingly felt that there is a lack of communication because we switched back to communicating science on virtual platform. So that's where we felt it. There is a lack of sort of a communications because we stopped attending international or national conferences, interacting with people physically. So this is where we felt we should understand the complexity of the subject with the leading experts. And we were extremely fortunate to have a series of wonderful webinar series. And today we have with us Professor Phil Kukura. Yesterday you heard him about the very, very basic fundamental of this single molecule spectroscopy. And today he's going to continue on that application aspects of those detection of single molecules. So with this brief introduction to this webinar series, now I will hand over to my colleague, Professor Jyotisman Dasgupta. Over to you, JD. Yeah.
Okay, thank you, Satish. And it's a pleasure to welcome, of course, Phil, and as I call him. I met Phil almost now 15 years back for the first time when I entered my postdoctoral work with Professor Rich Matthews at Berkeley. So there was a very brief overlap with Phil at that time. He was finishing up his thesis and moving on to his next big step, which was a postdoc at ETH Zurich. Of course, formally, would like to introduce Phil again. So Professor Phil Kukura is a professor of chemistry at the University of Oxford. He's also a fellow of the Exeter College in the university. He received his master's in chemistry from University of Oxford itself in 2002, after which he moved to University of California, Berkeley, and worked on ultrafast spectroscopy, especially developing femtosecond stimulated Raman spectroscopy to look at excited state structures of molecules in condensed phase. This was part of a big success that sort of motivated a lot more interesting areas of Raman spectroscopy in condensed phase. So this was one of the big tools that really showed that you could measure Raman spectra of time evolving molecules in femtoseconds to picosecond time scale. And Phil's PhD was a major contributor to this. After his really extensive ultrafast work, Phil actually went back to Europe and in ETH Zurich, where he worked with Wahid Sarnoter. And that's where he sort of started developing his clear interests in looking at extinction spectroscopy, label-free spectroscopy, which he started developing in the first talk yesterday. As we heard so beautifully, he developed it in terms of understanding the sensitivity of these tools as compared to what fluorescence already gives you. So Phil has been, after his postdoc and very successful postdoc at Wahid's lab, he moved on to the chemistry department in University of Oxford, first as a research fellow, then as a lecturer. And in few years of time, like 5 years, he became a full professor there in 2016. This rapid growth was, both in terms of his work in ultrafast spectroscopy, but also the development of the tool that he's actually now completely engaged in developing, in sort of applying in different kinds of areas, which is using these ISCAT as a technique to do imaging. Phil, for his wonderful work and creative work, has won multiple awards. That includes the Royal Society of Chemistry Harrison Meldola Prize in 2011, the Marlowe Award from the Royal Society, the European Biophysical Society's Young Investigator Award in 2017, and then again the Wolfson Research Merit Award in 2018. Very recently, he was chosen as the Blavatnik Award for Young Scientists in UK, which is one of the top prizes in Europe in chemistry in 2019, as well as the Medal in Light Microscopy in 2021. So with that brief introduction, I would like to get to Phil back for his second talk, where he will sort of look for mass calibration today. So I would just like to say to all the audience, it's refill time. So let's get this started.
To begin with the question, which I had in YouTube.
Yes, yes. We have Phil. Before Phil, if you could share your slides. But there was a question we could not take from YouTube yesterday. Could I read out the question? Maybe you can comment on it and then proceed.
You're on mute, I believe.
Yeah, of course.
Yeah. So this is from Arindam Ghosh. Great to hear about your science, always, Phil. Could one do intensity-based correlation with ISCAT using a prominent scattering probe like its fluorescence counterpart in FCS? I think you're mute.
Sorry, I'm trying to do two things, more than one thing at a time, which is always challenging. Can you repeat the question?
Sure. Could one do intensity-based correlation in ISCAT using a prominent scattering probe, like one does in FCS using a fluorescent counterpart?
I don't see why not.
Mm-hmm.
I mean, I guess you have these kind of, you have dynamic light scattering, multi-angle light scattering, right? So people use fluctuations in light scattering in order to find out about, diffusion coefficient of objects. So, that's a bulk method. I think if you were to do it like in confocal FCS, you would see the same thing. You would, in the absence of the probe diffusing through your confocal detection volume, you would see nothing. And if something comes through, you would see a spike. And then you could do the same kind of correlation time analysis that you do with fluorescence. So I see no reason why you could not do SCS, right?
Sure.
Scattering correlation spectroscopy. I think if you try to do it label free, then I think that would probably become quite tricky. Because you just don't have a lot of photons. I think that would be the major challenge, is how many photons do you actually get out?
Correct.
And I think, as I said yesterday, I think that's another one of those calculations I do about once a year.
Right.
And every year I realize that I have no photons. And no photons is a problem. So yeah, but it's a very good question, and I think the answer is I can't see why the answer would be no.
Correct. Okay, thank you, Phil. One more question, Phil. Just for sake, could you just go, if possible, in your slides, or maybe at the end of the talk you could do it. Just go to the detection part of ISCAT exactly, because there are some things that are not clear in my head. I know it's a simple idea, but it's still, I just wanted to know how you actually lock into your signal. So, or do you have a technique that enhances, because you're subtracting two things, and subtraction always leads to worse signal to noise. So than the original. So I just would like to get a little bit of a picture as to how actually you do it.
Do you want me to do that now, or you want me to?
No, you can wait till the end. Phil.
Okay.
If you,
okay.
yeah, let's move forward with this, and then at the end we can have questions and maybe go back to the ray diagram.
Just to ease your nerves. I mean, it's actually we're not doing anything. It's really just detection. But I'll talk more about that when I'm done. All right.
Sure. Cool.
Good. Right. So let me just do one thing. I did that yesterday. Just pull up the chat in case there are questions so I can see them. Right. So I hope I convinced you yesterday that you can do experiments that look a little bit like what I'm showing you here, right? You shine light on a single protein, and then you can see a signal that, if you like, you could consider to be a shadow of that protein. Something that is representative of the interaction between the incident light and a single protein molecule, right? And in our case, that molecule was binding on a surface. And now you might agree with me that if you had two proteins, the signal might be twice as strong. Right? That's not an unreasonable assumption to make. One protein makes shadow A, two proteins make shadow B, three proteins make shadow C. And the reason why that's important is, if that were true, what you would be actually doing is you would be measuring something that is representative of the mass, because two proteins weigh twice as much as one protein, very simply speaking. And essentially, that's the origin of everything that we have been doing with the technology since, is the realization that this does not just work in principle and in a cartoon, but in fact that you can use single molecule light scattering to very accurately measure mass and to obtain very high resolution. So where's the connection between mass and the interaction with light? So in this experimental setup where you're detecting scattered light from your protein and some kind of background light, which is, in our case, coming from the glass cover slip, your detected signal intensity is proportional to the polarizability of the object. And that kind of makes sense, right? The polarizability is essentially the ability of the electrons in the system to be driven somehow by the incident electric field. And if the interaction is strong, the polarizability is high. And if the interaction is weak, then the polarizability is low. And then you ask the question, okay, fine, what does my polarizability depend on? And the answer is, my polarizability is going to depend on the optical properties, which makes sense, right? Essentially, to which degree can the electrons be moved around? And it depends on the volume of the object, okay? Now, your optical properties will depend on the material to some degree, right? And the kind of, how the electrons are localized in your material. And proteins are all made of the same amino acids, right? And the electrons in amino acids are all localized to the amino acids. So you wouldn't expect the optical properties to really vary a lot from protein to protein. So this part of your polarizability term is going to be pretty much the same, whatever protein you look at. Then you think about volume. Well, so really simply speaking, proteins are all kind of the same size, right? Roughly speaking. I mean, if you've got a 50 kilodalton proteins, maybe 4 nanometers, 5 nanometers in diameter, and you could take 1,000 different 50 kilodalton proteins, and they're all roughly 4 or 5 nanometers in diameter. So the size, it's not like some are 50 nanometers and some are 1. The size does not change. And so what that tells you is that actually proteins all have very similar density, right? You get roughly the same amount of amino acids per unit volume for proteins. And that makes sense because you have common structural features, alpha helices and beta sheets and all of that stuff. So that kind of makes sense. So if that is true, given that your mass is equal to your density multiplied by the volume, if your density is constant, that means that your mass is proportional to volume. So if this term does not change, that means that your polarizability is proportional to the mass. So what we're measuring should be proportional to the mass. And this very simple picture that I've showed you before of two proteins should produce twice the signal as one protein should actually hold not only for one particular protein, but also for different biomolecules. All right, so is this true? So here's a movie of HSP 16.5. It's a heat shock protein, and it's quite special in the sense that it's highly monodispersed. It only exists in one form. It's a 20-former, and it has a mass of 396 kilodalton. If you run our experiment, you get a movie that looks something like this. All right. And you can see every single one of these black dots has exactly the same intensity. Yeah, all of them same intensity. If I run the same movie for HSP27, do the same experiment for HSP27, this is now a polydispersed protein, which means it exists as a dimer and as a tetramer and as a hexamer, as an octamer, as a 10mer, 20mer. You have all kinds of different oligomeric states. And of course, a tetramer weighs twice as much as a dimer and an octamer twice as much as a tetramer. So those should be different masses. And if you run this, now you see some of the dots are really quite dark and some of the dots are really so faint that you almost can't see them. So polydispersed protein, different signals, monodispersed protein, all the same signal. All right, so let's do another one. Here's BSA. So BSA, we're big fans of BSA, right? Because it's cheap and cheerful. There's lots of it. You can buy it from Sigma. And you run BSA, again, here's your movie, and you see sometimes you get a really dark spot, sometimes you have really faint spots that you almost can't see, and then some stuff in between. And what I'm plotting here is essentially the contrast, the size of the signal as a function of time for one of these movies. And for each molecule, I put one white dot. And where it is along this axis depends on how dark the dot is, and where it is on this axis depends on time. And you can see that clearly, you know, there are some bands. So there's a band here, and then there's another band here. And then if you integrate it up into a histogram, you see there's a third band here, and maybe even a fourth band over here. And in contrast space, you can see that they're equally spaced, right? So this is about 0.4%, 0.8%, 0.12%, 0.16%. And the obvious explanation is, you know, the first band is a monomer, the second band is a dimer, the third band is a trimer, and the fourth band is a tetramer. 1, 2, 3, 4. All right. And the mass argument holds that I've just told you before. So in a way, you could use these, you could find the center of this peak and the center of this peak and the center of this peak, and you would have a calibration. Because you know, this contrast corresponds to 66 kilodalton, this corresponds to 132 kilodalton, and this corresponds to 198 kilodalton. And now you have a conversion between mass and the optical contrast that you measure. So you do a calibration like this, and then you measure lots and lots and lots of different proteins, and you get a plot that looks like this. So this is the sequence mass of a whole bunch of different proteins ranging in mass from 40 kilodalton to 800 kilodalton. And this is the mass that we measure using a calibration like the one that I've just shown you. And you can see that, you know, it matches remarkably well. And if you look at the error that you have is on the order of a few percent. So, you know, 2%, is that good or is that bad? Well, it depends on how you look at it. If you're doing mass spec, then it's not very good. Mass spec can do a lot better. But for most cases, actually, it's not a bad thing, because what I'm telling you is if I have 100 kilodalton protein, on average, I will measure its mass anywhere between 98 and 102. It's pretty good, right? It's usually enough for me to go, oh yeah, I have the protein that I think I have, because I'm roughly measuring the correct mass. And Phil, these two, yeah.
Sorry, interrupting. These are in kilodaltons. You're showing the calibrations in kilodaltons. So 2% change in a 200 kilodalton protein would be quite a bit, right?
4 kilodalton.
4 kilodalton. So so we are off by at least 20 amino acids, right? Or 30 amino acids. Right. So is that going to be So for homodimers or homomers, in the sense of the same class, that is going to be nicely converging with your interpretation. But if there are mixtures, so in many cases, there might be slight differences in your monomers, but they will still interact and aggregate, for example. And would I pick up like changes there? If I'm, say, for example, out of 98 strand, out of 100 such mers, you have 2 or 3 which are distinct, which still aggregated because fractionally in the mutational space, there is only small changes in your protein sequence composition. So
no, I can't. I mean, that's where I'm in trouble, right? So I mean, I don't really have high resolution. So if I had a mixture of 98 proteins at 100 kilodalton, 98% at 100 kilodalton, and 2% at 98 kilodalton, I wouldn't even know. Right. I have no idea.
Right.
I wonder whether you would know with mass spec.
No.
And you might struggle, but
yes.
You know, for instance, there's another universe where this imprecision can also be helpful.
Right.
So a lot of proteins are glycosylated.
Right, for example.
And they're not all glycosylated in the same way. So you actually end up with quite a heterogeneous mixture. You try and put a glycosylated protein into a mass spec.
Right.
It's not a happy place.
Yeah. Right? So one opportunity.
But for us, it doesn't matter because we still just get, you know, we can't resolve.
Absolutely.
But we still go, oh, it's, you know, it's about 60 kilodalton.
So the mass spec specialists do it by a slightly more interesting manner. They do tandem mass spec, or they can do multiple sequential mass specs. So so that increases a bit of the information content. Maybe you know, a perturbation on these proteins separately can also be used as in your method. Maybe a perturbation of these aggregates could be used to sort of look for that information as well. Maybe, I don't know. I mean,
we're spending a lot of our time trying to think about how to implement approaches that are completely standard in mass spec, how to do them here, right? And you have to, you know, think slightly differently, but, you know, ion mobility is something you could implement here in a slightly different way. It's just not ion mobility, it's diffusion. So there's lots of games you could play in order to try and take advantage of some of the things that people are doing in mass spec. But, you know, at least from my experience, this has never been a problem. You know, I mean, Justin Benesh is my colleague who developed this together with me, and he's a native mass spec expert. You know, working on heat shock proteins, some of the most polydispersed proteins. Like he really, and he does co-assembly, exactly the kind of things that you talk about, 2 different monomers of slightly different mass. And he's kind of coming to the point where he says, well, you know, you need a pretty difficult problem in order to really be able to take full advantage of the capabilities of native mass spec if what you're looking at are proteins. That changes completely if you're looking at protein small molecule interactions, lipidomics. You know, there's 100 million things that mass spec can do that we cannot do, and we will probably never be able to do. And you know, it's just not within the capabilities of the technology. The big advantages is how it works. Right.
One more thing, Phil. More thing. It's a wonderful opportunity for polymer chemists to take, because see, the polymer problem of synthesis is it's polydispersed. For whatever method you use, and Satish will speak for it. Satish himself is a polymer chemist synthesizing these conjugated polymers in his lab, but he also knows how hard it is to get monodispersed polymers. So and the quantification of the polydispersity is something that I think you guys can do here.
So we've done some exploratory experiments in that regard, but then there is also the reality that, you know, you only have a research group of a certain size, and you have certain collaborators around you. There's only so much you can do efficiently.
Yeah.
But we have looked at the output of polymerization reactions, and we can actually see how the polydispersity changes as a function of reaction time. And this is all happening, but as always with collaboration, etc., it requires a certain impetus, and we're not quite there yet. But you're absolutely right. That's certainly one of the things that we could do.
Yep. Yeah. I think that's just follow up comment. I think it has implications in these all optoelectronic devices. People make it this out of conjugated polymer. You get so much difference in electrical and optical properties from batch to batch variation, and people do not have absolute control. So this whole lot of molecular electronics field, there is always a concern of lack of reproducibility. Every time you do, you don't see same thing. So that's again goes back to the fundamentally, how do you synthesize and how do you make this material in the lab, and do you have exact control on polydispersity? So that's very hard.
And the other thing I was always thinking, and to a certain degree that would also be true for inorganic nanoparticles, quantum dots.
Yeah.
Again, if you compare the ability to size individual particles here compared to something like using a TEM and drawing lines around particles, right? I mean, this is a lot more high throughput. You could do it in situ. There's lots and lots and lots of things you could play with, and I think there's lots of potential there. It just so happens that I collaborate with biologists, and I'm surrounded by biologists, so that's what we've been using. But I'm totally with you that the kind of synthetic universe is one that needs exploration. All right, so here we are, right? So we can, the two key things are, this is the ability to tell the difference between a monomer and a dimer. And that's incredibly important, because if you can't tell the difference, then you're doing effectively DLS, dynamic light scattering, right? But we can actually resolve these different species. So the resolution is a key thing, and the resolution originates in the signal to noise ratio, because it originates in the ability to measure the contrast for one molecule. That's what determines the width of these peaks. So you need to make them sharp enough so you can tell the difference. And of course, that enables, pretty much enables everything else that I'll be talking about. And the second thing is that your accuracy is good enough so that you have confidence that you know what you're looking at. If you have an antibody binding to a ligand, and the antibody is 140 kilodalton and the ligand is 60, then you really would like to have a peak around 200. And ideally, you'd see a peak at 140, a peak at 60, and a peak at 200, and then you know that you're roughly in the right space. So we published this now 3.5 years ago, and over the last 3.5 years, we've spent a lot of our time actually figuring out whether this is actually useful, right? For what can this be used? So one of the things was to look at the breadth, right? All the arguments I told you hold for proteins, but there's lots of other interesting biomolecules out there: sugars, lipids, nucleic acids. What happens when you go to a DNA strand? And on the one hand, you might say that's obvious. And on the other hand, you might say, I have no idea what's going on, because polarizability is a bulk property. What's the polarizability of a single DNA strand, right? And that's very different to a globular protein. A globular protein, you can convince yourself that the majority of the amino acids are a little bit like in bulk. But here you're really starting to get into problems, right? You have a molecular wire. What's the polarizability there? Then look at structure. And structure just from a perspective of, do I have something that is assembled, or do I not have something that's assembled, right? Because if these bits are missing, I have a different mass than if they are there. Interactions, I basically just said that if I can see this, and I can see this, and I can see this, I can determine equilibrium constants, because I can actually measure these things. And then membrane proteins, there's huge drug targets but a real pain because, of course, membrane proteins like to be in membranes and not like to be in solution. So you have to solubilize them in order to study them. Can you do mass photometry on membrane proteins? So I'll take you through these relatively quickly. So we'll start with the affinity. This was a nice little sketch that was commissioned by one of my grad students, but it's essentially that's what we do, right? We look at the proteins in solution and we can count. This is how many complexes we have, and this is how many free species we have. And if you can count all of this, then you can essentially read out the dissociation constant. So take something like tubulin. You have a dimer, and you have a dimer falling apart into two monomers. If you take tubulin and measure it at 20 nanomolar, you see two peaks, one at the monomer mass, one at the dimer mass. You can count how many dimers you have, and you can count how many monomers you have. You plug it in, and you get an equilibrium constant, get a dissociation constant, which is 8 nanomolar. And now you say, okay, fine, but surely you can't just measure this in 30 seconds, right? Is this actually correct? So what do you usually do to determine these things? You do a titration, right? So we do a titration, total concentration, 1 nanomolar, 2, all the way to 60. What you would expect at low concentration, below the KD, mostly monomer, at high concentration, above the KD, mostly dimer. And that's, you can see how the dimer grows in and the monomer shrinks. But what we can do is at each of these concentrations, we can do this counting, how many dimers are there and how many monomers. And at each concentration, we can determine the KD. And this is what we did here. And you do it on all of them, and then you measure that, you get the average KD. And again, it's 8.5 nanomolar. And then you say, okay, I'm going to do a traditional titration in terms of dimer fraction, then fit it. And from the fit, I get an equilibrium constant. And again, 8.5 nanomolar. So no matter how you do this measurement, you always end up at exactly the same. The other thing you can do is you can look at kinetics, right? I can either take my separate species, mix them together, and watch how my complex forms as a function of time, or I can mix them together at high concentration, dilute down, and see how they fall apart. And then I will get, I can get on rates and off rates. So people usually care about affinities, but they also care about on rates and off rates. And if you can measure the off rate and the on rate, you also get an equilibrium constant. So here we have something super tight, around 20 picomolar. When we do the measurement, we measure something very similar, around 50 picomolar, and similar for slightly weaker interactions. So you can get affinities and rate constants, which is what a lot of people care about. I've got a question here. Proteins flexible depends on the environment, hence the effective volume. As here density is taken to be constant, how do you use the calibration to detect same protein in different solvent environment where the effective volume could be different to different flexibility and partial denaturation? Right, so this is a tricky question. We're actually at the moment working on trying to really figure out what happens when you denature a protein. And it's one of these classic examples where you're in an office with a whiteboard. You're like, oh, we're just going to do this. We're going to denature the protein and measure the contrast and see how and then you do the experiment. It's a nightmare because really getting clean protein and then really knowing when it's denatured and how it's denatured. And if you use normal denaturant, stuff sticks. It's non-trivial. And then you want disordered proteins, and how disordered are they, and how does that depend on solution conditions? So you're absolutely right. The question is absolutely correct. It may well affect what we observe. But what's interesting is, even as I will show you just on the next slides, even the difference between DNA, the conversion between mass and contrast for DNA and proteins, I mean, they are massively different, right? Even that difference is not that big. And my interpretation of it is, well, biomolecules are all kind of the same. I mean, the reason why when you look at a cell, it just looks like snot, there's nothing there, it's because it's all the same kind of stuff. It's not like gold and rocks and water and, it's all kind of, it's organic stuff. And without much delocalized electrons, no response in the visible. So that's kind of all looks the same. And I think that's why we, roughly always get the same behavior.
Another question.
All right, so
sorry, Philip. Another question. if you were to think about the denatured state of the protein, you create it in a chemical environment of urea, for example, as in the buffer, or some cheotropic agent. So won't the optical properties and the refractive index now have to be normalized?
Exactly. There's a lot of problems.
Right.
You know, you add a lot of urea, now you change the refractive index of the solution, so you change the reflectivity of the interface, so then you change the contrast, but then your contrast change because you change the reflectivity, you know, does the urea bind to the protein? If it binds to the protein, does it change the mass of the protein? How much signal does urea produce when bound to it? You see, it just becomes a mess.
But your technique can address this age-old question, whether urea binds to the protein. That's really
and now because you're streaming on YouTube, I can't say anymore.
Sorry about that.
Okay, it's okay. Go ahead. Yes. Yeah.
Okay, so very quick look. You take a DNA ladder and you put it on, and you get this kind of movie. And again, you see some faint spots and you see some dark spots, which is what you expect because you've put a DNA ladder on. And here are your bands. And if you plot the number of base pairs versus the contrast, you get a nice straight line. And then you get a conversion between contrast and the number of base pairs. And you can do the same thing with single-stranded DNA. And now you get a straight line between nucleotides and contrast. And the ratio is slightly different. Yeah, and that's because the structure is slightly different. So that would be another way of answering the question. You know, here you have a significant difference between double-stranded DNA and single-stranded DNA. And indeed, you get a different conversion between mass and contrast, if you like. So it works with DNA, and the contrast is not that different. You know, the contrast to mass conversion is not that different to proteins, even though it's a very different material. And the good thing is that means that now you can do protein DNA interactions, right? Because you can see both of them. All right, the next one was membrane proteins, right? So as I said before, membrane proteins like to be in bilayers, so you have to solubilize them, put them in a detergent. And the problem when you do that is, you can see that quite nicely here. This is what everyone in cryo-EM that has the same problem, which is if you just put protein in a detergent and you stick them in the EM, all you see is detergent. Because there's a lot of detergent out there, in addition to protein within detergent. And of course, because we see everything, we will also see a lot of detergent. All right. So when we do this experiment, we see some dots due to our protein in detergent, but we also see a lot of detergent. But we can use the same tricks that people in cryo-EM use, which is you take a sticky detergent that really sticks to the protein, drop the concentration below the critical micelle concentration, all of your detergent falls apart, and all you're left with is protein. So you can see that here nicely. You take LMNG, dilute it, and you just get your protein complex detergent solubilized, and we see the same thing. LMNG produces a lot of signal. LMNG plus protein produces a lot of signal plus protein signal, and then diluted, you only see the protein. All right. And with that approach, you know, you can measure, for example, here you get the monomer versus the dimer of BO3. And of course, what we're measuring is we're measuring the mass of protein plus lipid, not just protein, because both of them contribute. And that's why the dimer is not at twice the mass of the monomer like before, because the dimer, you know, will have about 1.5 times the amount of lipid as the monomer, and it turns out that that's, you know, pretty much exactly the case. Where this gets nice is, you know, we had some collaborators who work on KCSA, and they measure the activity of KCSA using NMR, and they had 2 preps of KCSA. They make KCSA in these things called lipid nanodiscs, they're tiny little, you know, membrane mimics. And when you just run the size exclusion, they both look great. So the white and the yellow, they both look fantastic. All right. And when we took the same prep and we put them onto a mass photometer, we saw that the yellow prep had a really nice peak at 160 kilodalton, which is where you would expect KCSA in a lipid nanodisc to be. And the other one had a whole bunch of stuff that we didn't really understand. And sure enough, if you took the yellow prep and you put it in the NMR, you get all the peaks that you expect for a functioning KCSA. And if you took the white prep and put it in the NMR, you got nothing. All right. So even though on a sec column, they both look great, actually, once you can look at the mass with high resolution, you realize that one of the preps was good and the other one wasn't. And now you may understand why one of them actually is an operating channel and why the other one isn't. So then I mentioned structure, right? And of course, now we've got the cryo-EM revolution, right? You can get the structure of everything. And then maybe now we even had the protein prediction revolution. So maybe you don't even need the cryo-EM anymore, because you just put the sequence in and there's your structure, right? I mean, I'm not saying that I believe that, because you know, this is not my field, but there's one structural, you know, many structural revolutions. And of course, even for a cryo-EM to work, to give you an atomic structure, what you need is many copies of the same object, so that you can average them together. And in the averaging, you actually get the structure out. And that's really only going to work if, you know, a very significant fraction of the species that you have in your microscope are actually the one that you want the structure of. If it's too heterogeneous, you have a real problem. And if you have a multimeric species like this IPC complex here that consists of many proteins, how do you actually make sure that that is all there? So people put in huge efforts in improving their expression and purification protocols until they, you know, get a super clean sample that they can put into the electron microscope. So here's an example. This is a membrane complex that has a soluble domain and a transmembrane domain. And we stuck it, you know, did negative stain. And when you do it negative stain, you know, sometimes you see the whole thing. Sometimes you see a little bit missing of the soluble domain, and sometimes you just see the soluble domain. And in mass terms, again, we see the same thing. So here's just a soluble domain. Here's a little bit missing, and here's the fully assembled complex. So you can see, actually count the fraction of fully assembled species and see whether your prep is good. Sometimes people want the structure of something that is unstable. And so then they use cross-linking. So under conditions where there's lots of stuff, they put a chemical cross-linker, they, you know, glue all the little parts together and then purify. But how long do you cross-link for, under what conditions, etc., is not necessarily trivial to find out. But now, you know, here was a particular part of the nuclear pore complex, and they were interested in this complex 3. Under normal conditions, it was a minor contribution. Then they added 5 minutes, 0.1% glutaraldehyde, and you see suddenly that complex goes up. And you also see that it shifts in mass, which is good because we've added glutaraldehyde, right? It has more mass, right? Sometimes you have objects like, you know, here you have cohesin, which are structurally so flexible that it's really difficult to, you know, actually even figure out what you're looking at. And you know, do you have a good prep or not? So in the negative stain, well, there's something going on, tricky to figure out. You look at it in the mass photometry and you see, you know, you really have a dominant peak around 400 kilodaltons. So your prep is good, but it still may not give you a structure because, yeah, it's so floppy. And we then decided to really put this to the test in terms of, you know, how does this perform compared to normal protocols? And, you know, usually you have a whole purification protocol where you have different, you take different fractions and then you run gels and then you try and decide which one's the best. So this was for APC. So that's supposed to be at 1.2 megadalton, right? So you have a strep column and you look and you see, well, it's only a minor fraction here. And then you do ion exchange and you have 2 different fractions. This one's a lot better. And then you do sec and you say, oh, these 2 are really good and these 2 are not so good. And then for each of them, what we did is we determined the fraction with mass photometry, and then we did negative stain and determined the top classes. And sure enough, for these 3, the top classes were essentially all assembled complex, whereas for the others, they were something else. So there's a good match between what you're getting with negative stain and what you're getting with us, except that, you know, our workflow is probably a bit quicker. And then our collaborator also found some stuff in the freezer that they had from when they determined the structure, where they really purified. And sure enough, look at this, like, you know, absolutely crystal clean material. All right, so I spoke about membrane proteins before. And, you know, one of the issues is if you solubilize membrane proteins, and you're interested in protein-protein interactions, you have a problem, because that's not what they actually do. What they do is they interact in a membrane where they can diffuse freely and push lipids out of the way. Here they can't. They can't really do that. So the question was, is there a way we can do the kind of experiments that I've shown you, but rather than proteins binding to a surface and unbinding from a surface, do it in such a way where we're looking at proteins moving around? And if you think about the talk yesterday, I said that the key thing that you need is you need a change, right? Either a change because something binds, or a change because something unbinds, or a change because something moves. So you can subtract the background and actually see the protein of interest. And that's essentially exactly what Eric and Manish did. They implemented an algorithm with Gavin's help that essentially just takes a median background and can really remove the background, and all you see, the only thing you see is what moves. And when you do that with on a lipid bilayer on a piece of glass with dynamin, which is a membrane-associated protein, you get movies like this. And now you kind of see the same thing as before. You see some faint spots, you see some dark spots, but now they're moving around. Okay, so that means we can measure the contrast, but we can also measure the fusion. If we measure the contrast, again, we get these evenly spaced peaks that you saw before. So we can do a calibration of mass versus contrast, and now we can measure mass. We can localize these things with fairly high precision, so we can do tracking. But as we're tracking a particle, we can measure its mass the entire time, because the signal is proportional to the mass. We don't have bleaching, so we can measure for a long time. And if you can measure for a long time, you can measure the diffusion very accurately. But the kicker here now is that you have a two-dimensional measurement, because you're measuring mass, and for each particle, you not only measure mass, you also measure the diffusion coefficient. So here's a 2D plot of diffusion coefficient and mass, and you see the dimer diffuses quickly, and the tetramer is a little bit more slow, hexamer is even slower, octamer is even slower. And if you plot the diffusion coefficient versus the 1 over the number of subunits, you get a straight line. Similarly, if you plot the rate constant, so you saw some stuff binds and then unbinds. So if you essentially plot the residence time versus 1 over the number of subunits, you also get a straight line. What does that tell you? It tells you a dimer has 2 fingers in the pie, a tetramer has 4 fingers in the pie, a hexamer has 6 fingers in the pie, and an octamer has 8 fingers in the pie. So it actually tells you about how those oligomers are interacting with the membrane, and it gives you an additional way of differentiating species, because it's a 2D, it's a 2D spectroscopy, if you like, 2D measurement. And sometimes you could see the kind of things that we're ultimately after, where you may have a big object diffusing, and then after some time turns into 2, right? So you have dissociation, and sometimes you would have 2 diffusing around, and eventually, you have association. Now, admittedly, this is still a membrane-associated protein, not a transmembrane protein or integral membrane protein. But nevertheless, in principle, this works. There's an assay challenge, but in principle, we are now in a position where we can look at association, dissociation, assembly in bilayers. You have a question?
Yeah, a question. Very, very interesting. How many? So this will all depend on concentration. You will see the number of events that the bimolecular, say for example, dimers, folies to form a tetramer. So have you sort of figured out a way to look at, say, association constants on a membrane? Because these are hard to get from bulk measurements, and people have been trying to do that. And this is infinite time. You have infinite time to look at this, right? No bleaching. So have you actually given a full blow on these kinds of interactions, like you looked at these absolute on rates and off rates?
Yeah, you shouldn't be, it's the YouTube thing, Jyoti, is difficult. But the answer is yes, we're working on exactly those kind of things. And it works. I mean, it's challenging because it's quite new, and you really have to figure out, try and extract some parameters from it. And so there's a lot of modeling going in that we need to really understand the underlying thermodynamics. But you can essentially see it here, that's exactly what you can do.
That's awesome.
Right. And differentiate between different species. All right. And then I'm going to finish up just with one more story, and that is unpublished, but I think that's okay, where we try to look at actin. So, and it's a fundamentally a fairly simple question, right? You have proteins in solution, soluble, right? And there might be some monomers and dimers, and then something happens and it turns into a filament. And then something happens and it goes back. And of course, this is an essential process, for a lot of cellular function. And actin is only one example of many proteins that form filaments and fall apart, or form extended structures and fall apart. And so the question is, how does this work? What's the mechanism by which you go from left to right and back? And the main way of studying this has been ensemble methods, where you measure something like bulk scattering, or you have a fluorescence reporter that tells you when the filament is formed. And essentially you initiate the conditions where you should have polymerization, you have a lag time, then you have an exponential increase, and then the whole thing slows down. And what you do is you repeat the experiment at different conditions, different concentrations, temperatures, solution conditions, etc. And then you have all of these different plots, and you fit all of these plots, and from that you extract a kinetic model, and from the kinetic model you get your mechanism. It's kind of like chemistry a little bit, where is it an SN1 or SN2, right? You're extracting a molecular mechanism. The problem is, it's not necessarily just one model that fits, right? And of course, the issue that you have is you're doing this experiment, and the only thing you can see is the big stuff. You can't actually see what's in between. Now we were thinking, well, but we can see the little stuff, and we can also see big stuff, and we can also see stuff in between. So maybe we're in a position where we can actually monitor this process in real time. So here's some plots. This is actin below the critical concentration. So here, no filaments should be formed. And actin is 40 kilodaltons, so this is this main peak. Now you see this shoulder here, and you go, why is this a shoulder? There should only be monomeric actin. And the answer is, this is a log scale. Yeah, because we're a single molecule technique, we have huge dynamic range. So it's really difficult. These are only few particles, but we can still see that compared to what's going on over here. And you see, you initiate nucleation, nothing happens. Now you do the same process above the critical concentration, right? Okay, so you have a little bit more of these small species, but then as time goes by, you see how these here in this region above 400 kilodalton, you get real growth. You can clearly see species growing. So something is growing. This is good. This should be making filaments. And then what we did is we essentially, we modeled the distribution. So the green, I'm sorry, I'm colorblind. I should use better colors for myself. Is a simulation, and the yellow is what we have. And then we essentially run a kinetic model and see what happens. Okay. Now here's your traditional experiment that we discussed before, right? Plus the simulation with an existing model, and you see great, decent fit. Yes, I get filament formation. This is our view of the world, where we look at actually particles versus mass. And yes, we don't have enough statistics down here, but yes, filaments are growing. But when we look at the low mass region, we have a real problem. And the reason is that most of these models are nucleation-based models. So basically what they say is, in order for the filament to form, you have to make something like a tetramer or a trimer. And once you make the trimer, it either falls apart or it makes something bigger. So that species, those nuclei, are very low abundance because they're labile. Thus, nothing here, but we see lots, right? So we said, okay, let's take a different model. Yes, even worse, because that's what the model suggests, right? Nuclei are not there. Then we said, okay, we're going to ignore all the rate constants that everyone's ever measured, very dangerous, and we're just going to make it fit. Yes, perfect fit in the low mass, but now we don't make any more filaments. All right, so there was a problem. We couldn't get it to work. And then Nick is working with me. He said, I know what to do. I'm going to add more rate constants. There's a solution to everything, right? You can't fit the model. You add more parameters until it fits. And look at this, great fit to the bulk data, great fit. You see growth, filament growth, and low mass. And then you say, congratulations, you've added parameters and you made it fit. You know, anyone can do that, right? Well, here's a trick. So KB and KP are barbed end and pointed end on and off rates in terms of plus and minus. K hydrolysis is the ATP hydrolysis rate. And the rate constant that you see here, right? They're all from the literature. Every single one of them is from the literature. We only had to add one, which is the dimer falling apart into the monomer, because nobody's measured that one. And that was the only parameter that we used in order to make the fit. Everything else is from, you know, 15 years of bulk, very detailed, very good bulk experiments that measured all of these rate constants. So what's the key? The key is ATP hydrolysis. All right, because essentially, if you think about it, you're making these nuclei, they can fall apart, so they're always going to fall apart, but you have an on and off rate on both ends of the filament. If you have ATP hydrolysis and suddenly one end of your filament cannot fall apart as quickly as before anymore, what you're going to get is you're going to get net growth. Okay, so as far as we can tell from the modeling, it's the ATP hydrolysis that really makes these things grow. It's not some kind of nucleation-based mechanism, which, you know, kind of originates in condensed matter physics. All right, and with that, I'm kind of getting to the end of it. So I hope that I've convinced you that by shining light on proteins, you can measure their mass. And if you can measure their mass, you can do lots of interesting things. You can find out about structure, dynamics, mechanisms, interactions, purity. Just want to point out a few things, right? If you look at cryo-EM, you know, 15 years ago, this is what cryo-EM structures looked like. I mean, not great, right? Pretty blobby. And now they can see hydrogens. I mean, dramatic improvement. And why? Well, people worked on detectors and they worked on the electron sources, and they, you know, massive efforts in data analysis and sample preparation. And they went from this to hydrogens. And I think that this technology has a similar, you know, potential in terms of resolution, right? This is where we are. This is where we were when this paper was published 2 years ago. This is probably pretty close to the best instrument that we have in the lab today in terms of what it can do hardware-wise. Sample is a separate story, but hardware-wise, we're already here. And this is what I think is absolutely achievable, you know, with improvements in detectors and optics and data analysis, sample preparation. Now, the spectrum that I simulated here has 1 kilodalton resolution. Okay, this is 10 kilodalton. So at 1 kilodalton, you could have really complex mixtures. You can identify all of the species. You can see how they change as a function of time. And if you can do that, then you are really in a position where you can essentially just look at biomolecular mechanisms in real time, because you can tell what you have. You can tell the difference between different species because you measure mass. You know how much you have because you count particles. You can do the experiment dynamically, so you can you get information on change. And because you can identify peaks that are combinations of different species, you know what interacts with what. And those are really the kind of key questions that people have to answer in order to understand biomolecular mechanisms. And I think, you know, that's really the kind of the big picture, the kind of 10-year horizon here, which is going to be super exciting. And so with that, I'm done. Thank you for listening over 2 days. I want to say thank you to my group, because they are the ones who really do all the work. I just scribble stuff on the whiteboard. Lots and lots of collaborators without whom we'd just be studying BSA. I need to say a big shout out to Justin Benesh, who's, you know, had massive contributions to development technology. People gave me money, and once again, thank you for having me.
Great.
Fantastic. Well, like to please all of you unmute yourself. Give him a warm round of applause. Thanks, Phil.
Yeah.
Fantastic.
Wonderful. Now questions.
Questions, yeah. There are questions, I hope.
So anybody's I see here.
Okay, if the attendees who are not panelists, they have a question, please raise your hand. I will let you speak at that point of time.
Anybody has a question from attendees?
Okay, well, if not, then I have a couple of questions.
Yeah, go ahead. Yeah,
Satish, do you have?
Yeah, so it's just on a similar line of a thought. I think we had a brief discussion between about this conformational control of a protein or a polymer. It's extremely sensitive. I think you already covered that part. I think you already almost gave the answer. I'm repeating again, perhaps just to, how do we address that question? I think it's a very sort of kind of conformation these polymer or protein they adopt. It's extremely sensitive. So there's a diverse parameter which controls the conformational dynamics, and they're extremely sensitive. That creates a sort of a, in case of protein, functions is depend upon that, what kind of conformation they adopt. But if you draw the similar analogy in a polymer chemistry literature, there also you see a lot of these issues. How do we accurately determine the molecular weight? How do we accurately define the structure property correlations? So so your thoughts on that, little bit, maybe if you can elaborate on that, that would be great.
I mean, I think the key thing that we would need is a well-defined controllable system. That's really what's limiting us here.
Okay.
You know, if you gave me polymer A, and you could switch polymer A between extended polymer chain and crumpled up ball by changing something in solution that is relatively minor, then we could we could really begin to answer the question. I think that the thing that we've been struggling with is, once you get down to the detail, it's actually not that easy to switch between a completely unfolded protein and a completely folded protein in a controlled fashion.
Sure.
And I think to really be able to answer the questions, how overall structure affects polarizability and what we measure, we need a system that can be switched between two different states relatively easily.
Okay.
And I think that's the real kicker here.
Okay. Yeah. So JD, you want to?
Yeah. No, I think there are some very interesting directions that Phil, you are taking, and that's going to be huge in coming years. I guess one of the things that has been bothering many people over decades is, how does ATP hydrolysis lead to conformational changes, functional conformational changes? It's actually one of the big questions. If you could address mechanistically, of course you would require structure sensitivity. Just mass will not probably do it, but with additional sort of measurements, I guess what you can do right now is amazing, that in the real time, you could probably, by a model, a kinetic model, you can already see that there are nucleation mechanisms that need to be altered in active ATP hydrolysis machines that require active ATP hydrolysis. So my question is, are you thinking of expanding it with, or sort of making a hybrid with a fluorescence technique, which allows you to also get in real time some structural data beyond mass, which could help you in sort of disentangling this? The question has been for many systems, for the rhodopsin systems, for many membrane protein systems, there has been a lot of information from biochemistry that says the chemical energy generated from hydrolysis couples to the conformational change of the protein. The time scale of these are really not measurable. in most of the cases. It's just some idea that chemical energy got transferred into conformational change. Now that step is critical. It has to go through the vibrations. It has to go through the structural fluctuations. And so somehow that coupling step needs to be understood very well at a fundamental level. And I guess you're providing the first way to sort of hint at that by showing that acting polarization on an artificial membrane. But maybe have you thought of sort of looking at that question with additional tools and simultaneously what you can do with your mass measurements?
Yeah. So, I mean, I think, some of these questions, I think you can address with, if you have a fret labeled system and you add ATP and you see how the fret changes, you get information. I think what's really different, where I think that our real strength is, and we see this in a huge number of systems that maybe I wasn't able to talk about today, where you have a multimeric, multi-protein complex, and you add ATP, what happens? And the normal readout is, if I add this, I get high ATP turnover, and if I don't add this, I get low ATP turnover. And from that, and then here's a crystal structure, and I add these informations together, and I make an interpretation. But we can actually see what happens, right? We can see if you add ATP, this falls off, or if you add ATP, this comes on, or if you add, we can, because we measure mass, and if we have a 470 kilodalton complex and you add ATP and 60 kilodalton comes off, you're like, oh, well, that's what ATP hydrolysis does.
Right.
And if I have 410 kilodalton and I add ATP and nothing happens, I know, well, that's because that complex does not bind and hydrolyze ATP, and that's why you don't get any turnover. Right. So I think the real strength here, at least from my perspective, the real strength here is that, how do I say this? Biology is messy, and this, we have this, or many. How do I say this carefully? I think there's a tendency to see a crystal structure or a cryo-EM structure and say, this is it. This is what's doing everything, right? This is how those 7 proteins are arranged. This is what's happening. But that's not what you actually have, right? You have a dynamic thing that has on and off rates with stuff coming on and off, and those on and off rates are affected by lots of other things, either accessory proteins or salt or ATP concentration or conformational changes induced by something else. And I think that's where we really can add to the mix. In a single molecule experiment, again, if you have a multi-protein complex, you usually see 1 protein, maybe 2. But what about the other 8? Are they there? Are they not there? Right? And if half of your proteins are enzymatically active and the other half isn't, is that because one half is active and the other half isn't? Or is it because one half is fully assembled and the other one isn't? But you don't know, because all you see is what you've labeled.
Right.
So in my opinion, this is where where we have the possibility maybe to add the most benefit, is to really reveal the heterogeneity and the dynamics of these complexes. That's where I think we might be able to add something to all of the things that I already know.
And one more thing, there are preparations where you can take native membranes with complexes already there on top of it. There could be ways of. So have you looked at any of these natively biological membranes where you have these different, I know it's messy, I know it will be hard, but you have the mass labeling. So you could watch them and or could you simulate a biological membrane and put the components of, say, a reaction center, photosynthetic reaction center or something, and watch them come close and form it? So
I think the answer to both of your questions is in principle, yes. But I think we also need to kind of slowly ramp up the complexity, because you'd be surprised once you see everything. You'd be surprised by how heterogeneous even systems are that you think are super trivial. Like, oh, this is an antibody interaction with this protein, one-to-one, simple. And then you actually do it and you go, oh my God, didn't see this coming, right? So it's, I think what you've outlined very nicely is where we need to end up, but I think we still have a good long way to get there.
Wonderful, Phil. Wonderful. Thank you. Any other question? Yes, Diksha. Diksha is biologist. So.
Hello. So this may sound very naive, but I was wondering, in all the kinetics measurements, for example, when you are seeing what is the diffusion coefficient of bimolecular interaction, etc. So it will depend a lot on the concentration of the molecules that you have, because that will also affect maybe the diffusion coefficient. So I was wondering how much that will represent the true kinetics parameters that happen in the biological system, because there the concentration will be very different and their diffusibility will be very different, that can affect these parameters. So how are you selecting the concentrations that you are working with, and how is it representation of what happens in the biological system?
The dreaded physiological relevance question. The answer is, am I going to be doing experiments that are anywhere near what's happening in the cell? Absolutely not. It's a crowded, highly heterogeneous, highly dynamic environment. But then I would argue, 98% of all bioanalytical, structural, and biophysical techniques do not take place in the cell, right? So cryo-EM, not in the cell. X-ray, definitely not in the cell. Mass spec, not in the cell. DLS, any technique you want. SPR, BLI, ITC, I can go on and on and on and on and on. Everything has a certain concentration range. Everything has a certain environment. All of this stuff happens in an environment that is definitely not physiologically relevant. But I don't think that actually is, there is no, in my mind, there is no, there's no requirement for every individual experiment to be physiologically relevant. What you do is you obtain information on a particular system, and you take that information in the context of everything else there is, and see whether you can make sense of what's going on. So in other words, a classic example is if you want to understand transport and molecular motors, you can do a lot of in vitro experiments, right, which are not in a realistic environment, and you find out about the mechanistic aspects, how these molecular motors work. And then you compare the behavior with what you might be able to do within a cell, which does not give you that precision. But you see that maybe the processivity overall is the same, and you go, okay, it seems like roughly they're working the same way. So I think the answer to your question is, I don't have any hope that my measurements are representative of a true cellular environment, neither in terms of concentration, nor in terms of viscosity, nor in terms of the presence of accessory proteins. So for example, for actin polymerization, lots of accessory proteins, I don't have them in there. I can't recreate that level of complexity. But nevertheless, maybe my measurements, or the measurements that I've shown, maybe you think differently about how accessory proteins might work. The way people have thought about it is what the accessory proteins does is it stabilizes the nucleus, makes more trimers. But there's no evidence for that. It's just we're making an assumption that that's what's going on because that's what would make sense given the kinetic models from in vitro. Well, maybe what accessory proteins do is they accelerate or decelerate ATP hydrolysis, right? So, it's a big puzzle, and there's a lot of pieces, and there's a lot of techniques that contribute, and this is just one of them. But I completely agree to and concede that it doesn't necessarily happen in the conditions that that are representative of what's happening in the cell.
Yeah, okay. Thank you so much.
Thank you, Diksha. Any other questions? Yes, Philipp. Yeah.
Hello, sir. So I have two fundamental questions. I hope you can hear me.
Yes.
So, you showed a lot of movies like particles moving and all. So usually, is this always like spherical with this black and black color concentrated and with white color halo? Does it always be like this round spots, or you can actually tune the shape, like the point spread function that we like talk in some cases? Is it like that? Like I am not quite clear about this.
So if it is a subdiffraction object, then it will be just then it will just be that dot, because that dot is, you know, that's essentially the image that the microscope produces for anything that's smaller than 50 nanometers. You know, once you have an extended structure like a filament or something like that, you know, then you start seeing a sausage, right? But for everything that's small, you know, I would say up to virus size, it will always look exactly the way that I've shown you.
Okay, sir. Sir, one more doubt. Like,
Yeah.
if we have, say, more than one particle on, say, if they have one particle on another particle, so again, or like if the distance is different, like you see a particle, like one is much deeper than the other particle, will you get that contrast? Like, okay, one particle is actually sitting above the other particle and the other particle is on the glass slide or like that.
Mm. So information in Z in depth is quite tricky. I mean, in principle, the technique is very sensitive to it, but you end up convolving signal magnitude, and so it's not necessarily, you know, it's not immediately clear whether something is a little bit higher or a little bit smaller. Could be the same. So that's difficult to tell the difference. I think probably the best thing we have when it comes to 1 versus 2 versus 3 particles still is the mass. But how they are arranged in space, we don't really have much information, because, you know, as long as it is in the subdiffraction regime.
Thank you, sir. It was very wonderful talk.
Thank you. Okay, there is one question you can address in the chat.
Yeah, does wavelength. Yes, so wavelength, yeah, so the bluer you go, the more scattering you get. So blue is great if you want lots of signal, but the bluer you go, the more you blow stuff up, because you have more photon energy and eventually everything starts to absorb. So you have to kind of find the balance between getting lots of signal and blowing everything up. And that's usually, that we found that to be roughly indivisible. Could you increase spatial resolution? Well, if you think about it, I mean, we have movies where we have dots that appear and disappear, right? That's exactly the same as in super resolution localization-based microscopy. So you could localize all of these dots and then generate a super resolved image. But the question is, what are you going to image? You know, because we can't really do it in the cell because of all the background. It's really an in vitro super resolution, if you like. So we really haven't found a very good question in that regard, at least to date. But in principle, because you have dots that appear and disappear, you can use the same. You can use the same concept as in PALM or STORM or those techniques. Yeah.
Cool. Cool. Okay. Thank you, Devshankar. If there are no more questions, Satish and I would like to thank Phil for this wonderful set of 2 talks and really giving a fantastic start to our new year in the series. Phil, it would have been nice if you could have visited us for these talks in India, because you love Indian food, you also cook Indian food. So don't worry, this is not a missed opportunity. We will keep you in mind and we'll have extended invitation for you to come here. Hopefully we can organize a meeting for all the speakers at some point of time, actually a physical meeting.
That would be amazing.
Yeah, that would be amazing, because the speakers are fantastic. Greg Engel spoke, for example, in one of these sessions. So we have a bunch of spectroscopists, materials chemists, scientists. So it was fantastic. So thank you for joining in. And I would like to tell that we'll send you something on snail mail, something from our TIFR and IC part. So please look out for that.
Thank you.
Thank you.
Thank you
for joining.
Wonderful.
Thank you. Thank you.
Thank you so much.
Yes, so I think I will be just repeating what you said, perhaps on behalf of both the institute, actually. So we are indeed very, very grateful. I think, it was beautifully covered. I think in the first talk, in the way you started with very, very basic fundamental things, and that's where we want to convey our, for students. So it's basically that really worked very well. And indeed, we are very grateful. Thank you very much. And have a nice weekend.
It was an absolute pleasure. And I hope to see you in person in the not too distant future.
Yeah.
Thanks, Phil.
All right.
Thank you.
All right. Thank you very much, everybody. Cheers.
Yeah.
Bye-bye.
Bye.